Hello, I'm Yun-Soo Chang. What I'm going to talk about is a little understanding of liquid biopsies and genetic variations. First, to talk about liquid biopsies has come to the spotlight in a less invasive way for overcoming the weaknesses and limitations of the tissue biopsy, and for simple and repeatable reasons. Most liquid biopsies use blood, and there are difficulties in showing different sensitivity and specificity depending on different analysis platforms. It has been a decade since its release, and Professor Goto initially used serum to check for the genetic variations in the blood of 194 patients, when the sensitivity was only 43%. If other researchers used the plasma while studying to increase sensitivity, after deep consideration, the sensitivity increases by about 20% and PCR methods continue to be developed, such as Scorpion Arms, ddPCR, and NGS, etc. The liquid biopsy results using blood are 65 to 75% sensitivity. Although we'd think liquid biopsy is great through the press release, but there is a big limitation in that the sensitivity is not as high as we think. For this reason, circulating DNA, circulating tumor DNA, cell-free DNA, or cell-free DNA account for only 0.121.0, and the half-life time is only about 90 minutes. So the results obtained using blood and plasma are very less accurate than those obtained by tissues securing. Therefore, we questioned whether liquid biopsies using blood are the correct answer, or how to use other types of samples in liquid-type body fluid, and hypothesized that byproducts obtained during bronchoscopy can be used safely for liquid biopsies. Furthermore, we hypothesized that liquid biopsies using bronchoalveolar lavage would show a better variation detection rate than blood use. The subjects attended in the study are as follows. Total of 73 people were used to study, and there were 38 patients with early lung cancer and 35 patients with progressive lung cancer who could not be operated. 52% of people had the genetic variations commonly found in lung cancer, called EGFR, and 48% had no genetic variations. This can be seen as a form that reflects the subjects of general lung cancer in Korea. EGFR variated patients in Korea show more than half of all lung cancer patients, so I can tell you that this genetic test is important for this reason. This is an individual example. Here, blood is a blood-using test, and BWF is a ddPCR using a bronchial washing fluid, that is, a byproduct obtained when an organ performs endoscopy. If you look here, dark blue represents the coefficient of droplets, and green represents the coefficient of DNA variant. In other words, 439 variants were found in blood in the same test one, but 894 droplets, more than double, were observed in bronchial washing fluid. It is the same on samples number 2 and 3. The detection variation probability of is much higher when using bronchial washing fluid than when blood use. In other words, it means that we don't need to consider how much we're going to set the 3C hold, and the exposure probability decreases even though there's actually variation. And as an example, we get to know that there is no significant difference when you use the cell used as a positive control, called SNU 1330. In conclusion, I can tell you that bronchoalveolar lavage using is much more useful for variant DNA detection than blood use. In Korea, 90% of EGFR variations most commonly found in lung cancer patients are L858R variations or E19del variations. When checking whether there is a difference in detection rate according to this variation, only 47% of L858R can be found when detected in plasma on the blood, but the bronchial washing fluid shows an 89.5%, or nearly double detection rate increase. The second most common variation, called E19del, is the same. Plasma can only detect variations in 32%, but in bronchial washing fluid, variations can be detected in 68%, more than twice as many. If this is expressed through the ROC curve, it can be seen that the detection rate of variations obtained by the bronchial washing fluid is much easier even in individual cases. And when the two are combined, the variations detection rate obtained by bronchial release contraction is much higher. Also, if this is affected by variations, the sensitivity of plasma in early lung cancer patients is only 15%. But bronchial washing fluid can detect as much as 65% variations in early stages and 75% in advanced lung cancer patients who desperately need it. This is a very important result because depending on whether this gene variation is detected or not, treatable drugs vary depending on the condition of the treatment, and the prognosis varies depending on the drug. In the end, to sum up, samples using bronchial washing fluid can detect the genetic variations more accurately and sensitively than samples blood use in all cases, whether they are L858R, E19del, early or advanced. In summary again, we can say that detection of MEGFR by ddPCR using bronchial alveolar lavage is more effective than plasma-based liquid biopsies. So bronchial alveolar lavage is useful for avoiding related complications, such as invasive biopsy and pneumothorax or bleeding. Now the invention started from here. If so, what should be done to use this good bronchial alveolar lavage solution? There is a problem in that a sample must be collected from inner receptor, filtered out the rising liquid within 3 hours, but 1 hour ideally, and stored at -80 Celsius until the next experiment, or the experiment must be conducted immediately. The blood we used as a control group already has a tube. There are two types: Roche's DNA cell-free tube and Streck's DNA preserved tube, which makes it easy for us to think of as being applicable to collect the bronchial alveolar lavage. So, what was developed as a device as shown in Figure 1? The DNA breakage due to bronchial alveolar lavage is an important role in the breakage of secrete, blood, and white blood cells in the mucus. So, a device has placed the mesh on the upper part of a test tube that collects commonly used bronchial alveolar lavage to filter sputum and various cell components, and the lower part was developed by adding formaldehyde for DNA preserve and EDTA composition liquid addition to minimize the DNA damage. We devised this device because we did not use two different tubes to obtain the bronchial alveolar lavage filtered secretions of sputum and mucus, but needed an instrument easily to separate and collect one washing solution in one container and minimize DNA damage. Thank you.